Good morning, students. I am Dr. Uma Devi, Associate Professor, School of Pharmaceutical Sciences, Vistas. Today's lecture is about ICH guidelines. Let us see the contents. Introduction we are going to see, and ICH locations, objectives of ICH, the members, process of ICH, then the guidelines in quality, safety, efficacy, and miscellaneous. Now we will see the introduction. ICH stands for International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use. It's a joint initiative involving both regulators and research-based industry representatives of European Union, Japan and US in scientific and technical discussions of the testing procedures required to assess and ensure the safety, quality and efficacy of medicines. These are three pillars on which the health of patients depend. ICH is located, the Secretariat is based in Geneva, and the biennial meetings and conferences of ICH Steering Committee rotate between the European Union, Japan and USA. Now we will see the objective of ICH. To increase international harmonization of technical requirements to ensure the safe, effective and high quality medicines are developed. To harmonize the technical requirements for registration or marketing approval. To develop and register pharmaceuticals in the most efficient and cost-effective manner. To promote the public health. To prevent unnecessary duplications of clinical trials on humans. And also to minimize the use of animal testing without compromising safety effectiveness of drug. ICH members. ICH members, it's comprised of representatives from 6 parties that represents in Japan. The members are the Ministry of Health, Labor and Welfare and the Japan Pharmaceutical Manufacturers Association. In Europe, the members are the European Union and the European Federation of Pharmaceutical Industries and Associations. In the USA, the members are the Food and Drug Administration and the Pharmaceutical Research and Manufacturers of America. Additional members include observers from the World Health Organization, European Free Trade Organization and Canada. The observers represent non-ICH countries and regions. Now we will see the process of ICH. So different steps we have. The first one is draft are prepared and circulated through many revisions until a final harmonized draft is completed. In step 2, this draft is signed by the working group as the agreed upon draft and forwarded to the steering committee for signing, which signifies acceptance for consultation by each of the 6 co-sponsors. In next step 3, the 3 regulatory sponsors initiate their normal consultation process to receive the comments. In step 4, it is reached when the steering committee agrees that there is sufficient scientific consensus on the technical issues. So this endorsement is based on the signatures from the 3 regulatory parties to ICH affirming that the guideline is recommended for adoption by the regulatory bodies of the 3 regions. So further in step 5, the process is complete when the guidelines are incorporated into the national or regional internal procedures. So now it is implemented in the 3 ICH regions. So now we are going to see the ICH guidelines in the aspects of quality, safety, efficacy and multidisciplinary area. In quality guidelines, so the quality area include a vital milestones such as conduct of stability studies defining relevant thresholds for impurities testing and a more flexible approach to pharmaceutical quality based on good manufacturing practice risk management. So in safety guidelines, a comprehensive set of ICH guidelines that help identify the potential risks such as carcinogenicity, genotoxicity and reproductive toxicity. So in the efficacy guidelines, it's concerned with the design, conduct, safety and the reporting of clinical trials. It also covers novel types of medicines derived from biotechnological process and use of pharmacogenetics genomic techniques to produce better targeted medicines. In multidisciplinary guidelines, there are three cross-cutting topics which do not fit uniquely into one of the quality, safety and efficacy categories. It includes the ICH medical terminology, the common technical document, and the development of electronic standards for the transfer of regulatory informations. Now we will start with the quality guidelines. Stability, which starts from Q1A to F. Q1A are 2. Stability testing of new drug substances and products. This guidance is for analyzing the product to determine its stability in a variety of environmental conditions. Q1B, stability testing for photostability testing of new drug substances and products. Also, for stability testing, this guideline studies the effect of light on farm products. Q1C, stability testing for new dosage forms. This is on annexure to the ICH harmonized tripartite guidelines on stability testing for new drug and products. Q1D, bracketing and matrixing designs for stability testing of new drug substances and products. This applies to stability testing of a product using bracketing and matrixing. Q1E, evaluation for stability data. This section addresses the method of evaluation of data after the analysis of drug products for stability testing. Q1F, it's for stability data package for registration in climatic zones 3 and 4, explanatory note. Now we will see the analytical validation. Q2R1, it's a validation of analytical procedures, text and methodology. Q2R2 in Q14, analytical procedures development and revision of Q2R1. Regarding the impurities, Q3A is there in R2, impurities in new drug substances, Q3B1 also is given. Q3B R2, it's impurities in new drug products, Q3C R6, residual solvents, Q3D, it's elemental impurities. Regulatory acceptance, ICH Q4A to B, pharmacopoeias is given. Now we will see the quality of biotechnological products. It's under ICH Q5A to E, quality of biotechnological products. Specifications, ICH Q6A to B, it's talk about the specifications. Good manufacturing practices, Q7, good manufacturing practice for active pharmaceutical ingredient. Pharmaceutical development, Q8 or 2, it's for pharmaceutical development. Q9, which discuss about quality risk management. In Q10, pharmaceutical quality systems. Q11 Development and Manufacture of Drug Substances. Life Cycle Management ICH Q12 Technical and Regulatory Considerations for Pharmaceutical Product Life Cycle Management. Continuous monitoring of drug substances and drug product. In Q13 Continuous manufacturing of drug substances and drug product. Analytical procedure development, it will comes under Q14 analytical procedure development. Now we are going to see the safety guidelines. It starts with S1A to C for carcinogenicity studies, and S2 which discuss about genotoxicity studies, S3A to B for toxicokinetics and pharmacokinetics. And S4 is the toxicity testing, S5 is the reproductive toxicology, S6 which tells you about biotechnological products, S7A to B is connected with pharmacology studies. In S8 immunotoxicological studies are given, and S9 non-clinical evaluation for anti-cancer pharmaceuticals. In S10 photo safety evaluation, and S11 non-clinical pediatric safety. In efficacy guidelines, E1 which discuss about clinical study for drugs used in long-term treatment. E to A to F pharmacovigilance, and in E3 clinical study reports. E4 is the dose response studies, E5 its ethnic factors, E6 good clinical practice, E7 clinical trials in geriatric populations, E8 general considerations for clinical trials, E9 statistical principles for clinical trials, E10 choice of control group in clinical trials, E11 and E11A clinical trials in pediatric populations, E12 clinical evaluations by therapeutic category, E14 clinical evaluations of QT, E15 definitions of pharmacogenetics, pharmacogenomics, E17 multi-regional clinical trials, E18 genomic sampling. Now the last one is multidisciplinary guidelines. So M1 is the MedDRA terminology, M2 is the electronic standard, M3 is non-clinical safety studies, M4 common technical document, M5 data elements and standards for drug dictionaries, M6 gene therapies, M7 mutagenic impurities, M8 electronic common technical document, M9 biopharmaceutics classification system based biowaivers, M10 bioanalytical method development. These are the references. Thank you students for your patience hearing.